Good morning, everyone. My name is Monica Jean. I'm a field crops educator based in the Central Michigan region. So right now I'm sitting in the Isabella County office, and I'm the host today for Dennis Pennington, our really awesome wheat specialist. And we're hoping to have a fun program today, learn about wheat, learn about the weather, and then of course come with all of your questions at the end. So hopefully we can have a great conversation. I would like to introduce to you Dennis Pennington. He is our wheat specialist here on campus working with the Michigan Wheat Program. And I would like to say he does an exceptional job. And we're happy to have him on today to talk about heat stress and wheat. Hey, Dennis.
Good morning. Well, thanks for the introduction, and I'm not always that great to work with, or at least I don't think I am. So, but all right. So, good morning, everyone. I'm Dennis Pennington, Wheat Extension Specialist with Michigan State University. Um, I thought this might be a timely topic as we're moving it toward the end of May into June. But as you know, the weather pattern has been a little different than what we would call heat stress. But we'll nevertheless, I put some slides together to learn a little bit more about heat stress. So we'll go ahead and get started on that. So what is heat stress? Heat stress refers to the physiological and biochemical processes or disruptions that occur when wheat plants are exposed to high temperatures that exceed their tolerance level. So there's a number of different things that happen, both physiological and biochemical. What is the optimum temperature for wheat? It's 54 degrees to 72 degrees. So if you think about where we have been for the last week or so, we've been right in this optimal range. And so despite the cool temperatures we've had, the wheat is still actively growing. So now these disruptions start to occur when the temperatures get above 88 degrees Fahrenheit. And when temperatures get that high, we see reduced fertilization, which results in lower grain numbers and deformed kernels. Now, in Michigan, this usually occurs during the grain fill period, and the grain fill period can be identified as from the flowering date until physiological maturity. That is the grain fill period for wheat. Typically in Michigan, that is 28 to 32 days, depending on the season. Once in a while, we'll get a longer grain fill period than that. So if you just take an average of a 30-day grain fill period across the state, and then compare it to some research that shows that the grain fill period can be shortened by as many as 3 to 12 days. So if we have those high temperatures during that grain fill period, even on the low end, even 3 days, three going into 30, that's 10%. So it can cause a significant impact on yield. And the extent of the impact is going to depend on how high the temperatures get, and then the duration. Is it just for a few hours or is it a few days? And then something that sometimes is overlooked in the research is what's the impact of the overnight low? Because sometimes when we have those 90, 92, 94-degree days in the evening, it might only get down to 70 or 72. And at that point, we're just down into the normal range. So the wheat doesn't get much chance to rest and recover overnight when we have those high overnight temperatures. So to help understand what's going on in the plant, let's review some basic plant biology. So if we look at the cross section of a leaf of wheat, you've got the upper cuticle here, and then you've got upper epidermis, you got the palisade mesophyll cells, and then the xylem and phloem is here. This is what moves water and nutrients and solutes, the sugars that are produced through photosynthesis. And then, of course, you got your lower epidermis, and then the lower cuticle on the bottom side of the leaf. But heat stress in particular plays a really important role in these guard cells. These guard cells are what make up the stomates, and the stomates are what allows for gas exchange. So if you zoom in on these guard cells down here, this is what it looks like. And this is a typical diurnal pattern. So when the guard cells are open, the stomates are open. This happens every morning when the sun comes up. And then you get this normal gas exchange where CO2 is taken up by the plant, and then through photosynthesis, it releases oxygen, and then water is respired through the plant just for normal cooling of the plant. Then in the evening, when the sun goes down, these guard cells close, and then that maintains the plant integrity and whatnot over the evening. So what happens when you have heat stress is the these guard cells, typically would be open during the day and doing this gas exchange here. But when you add that high heat, these guard cells can close, and your stomates will close even in the daytime as well as the nighttime. And then so the gas exchange basically has stopped. So if you remember back to high school biology, photosynthesis, plants utilize light energy. They take up carbon dioxide, and through photosynthesis, they produce carbohydrates. They release oxygen, and they release water back into the atmosphere. This water is released just as the normal cooling or evapotranspiration that occurs in the plant. Now, this arrow is pointing the wrong way. I created this image in Chat GPT, and I could not convince it to turn that arrow around. So it's the water is not what the water does, or the plant does need water, but this arrow is supposed to show that it respires water. That's part of the normal photosynthesis process. But when you have this high heat, that gas exchange basically stops. So it's a bit complicated. There's a lot of things that are going on in the plant. In order for researchers to try to create mechanisms or ways to deal with or manage this stress, we got to understand these. So photosynthesis is one of the key things that is affected. There's a number of different plant processes that are changed with high heat stress. Carboxylation is decreased, rubisco production is decreased, the activase enzyme is decreased, photosystem 2 is reduced, chlorophyll production is reduced, phytochemical efficiency, photorespiration goes up. In terms of leaf senescence, we start to see changes in the plant where digestive enzyme production is increased, lipid peroxidation is increased, and all those things lead to senescence of the leaves. Respiration will increase, root and plant respiration when under heat stress. And then there's oxidative stress things that happen, like reactive oxygen species production increases. You get accumulation of stress metabolites, destruction of plasma membrane, leaching of electrolytes. And when this stuff all happens, now it throws off your water relations. So your turgor and osmotic pressure, that is decreased, and your leaf water potential is decreased, which leads to wilting of the plant. And when you have all of these things going on, of course, the transporter system for nutrients and nutrient uptake is also decreased. So there's a lot of things going on in the plant that affect normal plant operations, and there's some key growth stages when this occurs. The amount of damage to yield is different. So the timing of the heat stress matters. So let's talk about the pre-flowering and flowering stage. This is heat stress during this period is when you'll have the greatest impact on your yield and have the greatest yield reduction. So there's a critical period. And this critical period is defined, or what occurs during then is the number of seeds per spikelet is being determined. So when does this occur? This period lasts from about 20 days prior to flowering to 10 days past flowering. So when I think about where we are in Michigan right now, most of the southern half of the lower peninsula, we are at flag leaf to even head emergence. And in the south, we're actually close to flowering now. So we are in this critical period. So the number of seeds per spikelet are being determined right now. And this is a 30-day window. So this is a pretty wide window where if we have heat stress, we can have significant yield loss. One of the things that is affected the most with heat stress is pollen formation. This begins 5 to 7 days prior to flowering. and then it extends 3 days past flowering. And this particular stage is really sensitive to heat stress. So, I'll show you in a minute here the kind of a zoomed in picture the flower and how fertilization occurs. But when you have heat stress during this pre-flowering and flowering stage, um, you get a lower grain number per spike. You just have less seeds in the head. So let's look at these pictures a little bit more closely. This is a spikelet. You can see in each spikelet, there's the male parts are the anthers up here, and then these feathery looking things are the stigmata. Those are what receive the pollen from the anthers, and then pollen tubes must grow down into the ovary in order for the pollen to fertilize the ovary to get a seed. So let's look at an individual floret a little bit closer. And you can see pollination is going on in here right now. Here's your anthers. It's releasing pollen in here. The pollen is landing on the stigmata inside here. And then that pollen has to travel down this tube. So here's a zoomed in view of this tube. So not only do we have to have the pollen grains be formed, so heat stress will reduce development of the pollen grains. Heat stress can also disrupt the development of this tube. So if this tube isn't developed, even if you have pollen grains, if they can't get down in here to the ovary, you won't get a kernel, and you have basically an infertile floret in your spikelet. So when you look at what's going on in the field and you look at this head here, you can see the anthers are being extruded right now. We're not at this stage yet in the field here in Michigan, but in these two spikelets right here, this flowering has already occurred. So pollination and fertilization has already occurred in these two spikelets once you see these anthers on the head. And this is also a critical time because this is the trigger for when half of the heads in the field have these anthers coming out. That's the time to make your fusarium head blight application. So just know that when you're starting to see these anthers, the fertilization has already occurred inside the heads. So or inside the individual florets. So if we look at post flowering, we get outside of that 30-day window mentioned up above. There is an impact to yield, but it's a much lower impact. And usually what happens here is you'll get abortion of grains, decreased grain weight, a little bit higher leaf senescence, and some grain quality reductions. So bottom line, it's complicated. There are a lot of processes that occur in a wheat plant, and this diagram is showing a bunch of those processes and what happens in the plant when you have heat stress. So what's being done about it? Well, there has been some breeding efforts for heat tolerance. There's been some QTL clusters that have been found on these different chromosomes in the wheat. There's been some GWAS or genome-wide area association studies. For example, one of them found that ethylene production in the spike actually decreases the spike dry weight. Ethylene is a plant hormone typically associated with ripening. So that's an indication. What this means is that under heat stress, ethylene production goes up, and then your spike dry weight goes down, lowering your yield. Phenomics is another tool that breeding programs can use. This is looking at the phenotype rather than the genotype. Things like leaf chlorophyll content or chlorophyll fluorescence are things that can be used to identify QTL that then can be used to develop new lines. And as you can see, there has been some success with creating heat tolerant lines, but we're still really early in the development of this, and we've got to have a little bit more time to get to where we have locally developed or adapted wheat lines that have this heat and drought tolerance. So what can farmers do? Basically, the best thing you can do is grow the best crop you can. Follow good husbandry practices that promote good root development, improve your soil structure and soil health, things like eliminating compaction, managing diseases, incorporate cover crops anywhere you can in your crop rotation. Make sure you maintain good crop nutrition. And then I would also like to encourage on-farm testing. The only way you're going to, you know, identify what are the factors that are limiting yield on your farm is to do strips with and without a product or one practice versus another. Do them in the field and take them to yield and compare the yield so you know and then fix whatever you can fix. Once you've identified a yield limiting factor, fix it. If you can do all of these things on your farm, you should have a crop with a good, healthy above ground biomass and a healthy root system that will help you to get through a heat stress period. So there's still a lot of work to be done in the area of heat stress, but hopefully this has given you just a little bit of information about kind of the processes in the plant are kind of complex, and so coming up with an answer is going to be a tough situation to try to figure out. So, Monica, I think with that, I'll end here.
Yeah, great job. I don't think you went that far over. So
Okay, good.
we're gonna make it then to the chat box here. We had quite a few questions start rolling in. Dennis, do you just want to start at the top and we can work through? So the first question I have is, what is a QTL cluster?
If you're not in the plant breeding world, you might not know what that is. And I am not a plant breeder, but I work with one. So, QTL, so when you're trying to breed for specific traits, let's say we identified a variety that has higher gas exchange during high heat time frame. We then need to try to figure out what DNA is different in that plant. What is the genes encoding for that uh, that allow that plant to tolerate that and still allow that higher photosynthesis that might be going on because of that. So QTL is a tool that allows you to take these measurements and try to identify segments of DNA. You're still not down to the gene level yet, but the QTL is a section of code in the DNA where you think that gene resides. And then there's other analysis like GWAS that can help you narrow that down further to try to identify what are the specific target genes. And then if you can identify what those genes are, then you can screen all of your germplasm that you have, find out which varieties have those particular genes, and then that would be a target for breeding efforts to try to make crosses. So that's maybe getting into the weeds a little bit in that. It's a bit complicated. Eric Olson's our wheat breeder here at MSU. He's very good at doing this stuff. And so he could probably do a better job explaining QTL than me. But okay, so
And then the next one is about that 88 degrees Fahrenheit, if it's magical.
yeah, Peter, that's a really good point. I don't think 88 degrees is a magical number that you're automatically going to have the same amount of heat stress and yield reduction. Um, it really depends on the crop going into it. If you got a crop that is diseased or has been lacking in nutrients or for some other reason is stressed, or maybe there's a compaction layer that's causing roots to not grow deep, anything that can cause stress in those plants, they might have a more significant yield reduction at that 88 degrees or perhaps even a little bit lower temperature, whereas a plant that is healthy, free of disease, got adequate nutrition, it's got a good root system developed, you got good soil moisture conditions, that plant could probably tolerate a little bit higher temperature. But this 88 is what's used in the literature as kind of the cutoff that where you get above that and you can start to see those problems. So,
Right. The next one on here is about applying sugar and foliar for growth and yield. If that helps, if you're aware of any studies that show the effects. And besides wheat, is it known to benefit more than just wheat crops or what's out there?
yeah, so there is a little bit of research on a couple of different things, sugar being one of them. Silicon is another one. that if you apply it prior to or ahead of the stressor, the heat, like if you got a 92 degrees in the forecast for tomorrow, if you could make an application today, that will help the plant manage photosynthesis and manage water relations a bit. If you can keep those stomates open during the day under that heat stress, as long as you got adequate soil moisture, those products will help the plant get through it. Now, there's a point at which if you have 4 days of 90 degrees higher in a row, there's a point at which it's too much that these things won't help get through something like that. But there is some work out there. I haven't done as much digging into the published research in that area. But yeah, I will be doing a little bit of work with silicon on. I'm hoping to have during our grain fill period here a high degree day where I can do some testing with silicon this year.
And Jenny was hoping you could elaborate a little bit on the ethylene effects on test weight and if there's
Yep.
management that can be done to increase that test weight.
Yeah, so I wonder if this question is specifically about heat stress or is this about just increasing test weights in general? Under heat stress, yes, that one research paper I mentioned studied and found that you get higher under heat stress, you get higher production of ethylene in the spike. And what that causes is a reduction in the size of the kernels. So the spike weight was decreased. Um, and what that indicates is that, the kernels just basically didn't fill completely because the plant shut down and matured, prematurely, which would lower your test weight. So, anything you can do to keep your grain fill period going longer, make sure you got adequate nutrients out there, healthy root system. Your goal is to extend that grain fill period as long as you can. Fungicides will help do that. Um, so, yeah, in the case of heat stress, yeah, you can get reduced test weight simply because the grain fill period gets cut short kind of abruptly.
All right. And then
Then
the next one coming in is from Phil. Yep. About how, if we have high temperatures, how long do those temperatures have to exist in order to affect plant physiology? As an example, if we hit 90 for an hour or two and then decrease to mid 80s, will there be a decrease in pollen and ultimately yield?
So I think if it's just a short, you're above 90 for a few hours on one day and then it cools off in the evening, the effect is going to be minimal, even if you're in the flowering phase. Because as you know, flowering goes on over a period of time and fertilization goes on over a period of time. Not all heads flower exactly on the same day. Not all spikelets on the head flower at the same time. The flowering and fertilization starts in the middle of the head and then progresses to the distal and basal parts of the head from there. And then tiller heads might be a few days later. So I mean, yes, it could have some impact, but it sounds to me in that scenario it would be a lower impact on yield than having, a day or two in a row. And then if it cools right off in the evening too, that will help. But
Oh,
it's really hard to gauge how much yield loss it will be. It really depends on how healthy is the plant going into the heat stress, because
okay.
a healthy plant will be able to tolerate much better.
The next one's about an interaction between heat, street, stress tolerance, and plant available soil P.
So I haven't looked specifically at plant available soil P and how that relates to heat stress. But I think it still goes to the fact that if you have a plant that has a stressor, like lack of phosphorus, that can increase the risk of heat damage, making or causing greater yield loss. Um, so I don't know specifically about soil test P related to heat stress. I see Peter Johnson's on here. If Peter, if you have any experience with that, I'd love to have you turn your microphone on and respond. But I don't have anything specific on that.
Yeah, so there's if the plant's under stress, I mean phosphorus, we need some it's active uptake by the plant. If you're under stress, you're likely to have less photosynthate available for active uptake. But there's no relationship that I've seen specifically in terms of heat stress tolerance, like a plant that is more tolerant to heat and its reaction to soil P. The only reaction would be in that amount of photosynthate available for phosphorus uptake.
Yeah.
Awesome. Thanks,
Thanks, Pete.
Peter. Thanks for being on too. All right. I think next Peter asked a question actually, and it was to Jeff about any 88-degree plus temps in the next 35 days that could spoil the wheat grain fill
Right.
period.
And the response, I don't see this happening anytime soon, but given the outlook and that medium range, especially as we move into the latter part of June, it's certainly possible. It's not likely for most of our area. 88 is a pretty warm day. So again, you can never say never, but it's very unlikely for the next at least for the next couple of weeks. But maybe after that, there could be a chance. But right now, don't see any major heat waves yet, anything like that showing up. It always have to watch and things change, but right now, at least for the next couple of weeks, that does not look likely.
Okay, and since you're on here, Jeff, like we got you talking, you want to answer the wind question also? Will they continue
Ye-
to be higher than normal?
Yeah, most of these strong winds that we see, they're associated with that upper air pattern, those big cut off low features and what goes on the surface, it's all related. And the more of those we have, the stronger the winds are at the surface. So some of it is just, and that of course it just happens. It's not typical, but it happens once in a while. And so most of these really windy days we've had are because of that. We've looked at data uh, for a long time for the wind, and we can't really identify any long-term trends with it's difficult to do. There's a number of reasons for that, but we just don't see anything significant one way or the other. So that I would also that's an important, I think, context is here as well. But recently, look at the jet stream, and ultimately that's why we've had some of those strong winds on the surface as well, that pattern.
All right, and then back to your comment about the temperature, Peter is confirming record wind fields will
There we
occur.
go.
So thank you for that. I think we're going backwards a little bit, Dennis, here. The previous question about sugar,
Yep.
David had a follow-up here. Does
Yeah.
applying before, during, or after times of stress help the plant in any way, aka physically, chemically?
Yeah. So I got to warn you, I have this software center thing loaded on my computer from because it's owned by MSU. It's going to force a reboot in 4 minutes and 1 second.
Well,
So
you better talk fast then.
So I just wanted to make you aware of that.
Okay.
I tried to cancel it and pause it and but now it won't pause anymore. Um so back to the sugar question. So my understanding of the sugar, the silicone, the tools try to help get through a heat stress problem, you need to apply it just ahead of when the stress occurs. One of the things that I'm interested in finding out is how long does an application last? How long do you get an effect from application of that product? Is it only good for a day or two, or does it provide you some lasting help in terms of heat stress? And so I don't have answers on that. I've tried to look in the research, but certainly you would want to apply it ahead of when the stress is. If you try to apply it after, you let the damage already occur. So that really wouldn't help you.
Okay.
Um,
There was
and
one
then
question too. The fungicide on wheat in Southeast Michigan, and just so you know, in our wheat watchers, we've got Feeks 10.1 and 10.3.
Yeah. Yeah. So, Paul, I would say that I would go out and look for anthers in your field. I'm assuming you're talking about the T3 flowering fungicide timing. Your goal would be to hit that after flowering has started, not before flowering has started. There is a fungicide product that's labeled for 10.5, which is when the head is fully emerged from the boot. But we tend to get better efficacy on DON control or vomitoxin control when we still make that application at the flowering timing. So yeah, I would definitely go out and look at the heads and try to hold off. I know you got to play the weather, but make the application when you're seeing flowering in the field. So beginning of flowering would be when half of the heads you can find anthers on them.
Okay, that's all the questions I have right now, which might be perfect since your computer's going to reboot.
45 seconds.
All right. Well, I'm just going to remind everyone that, of course, we're on next week talking about dry beans. This is available if you want to go back and review anything in a podcast. And I think because of your issue, we're going to move towards other specialist updates on here right now. So, anyone else that is
No one's saying anything, so I will say something.
awesome. Thank you.
Black cutworm trapping is sort of trailing off, sort of ending. True armyworm, that data is in the Great Lakes and Maritimes Trapping Network. I forgot exactly what it's called, but if you search for it, it'll come up and you'll see
I'll
that
pull it up.
information. Yeah, and then so then we will switch over to Western Bean. And people have asked me when do I start? I usually start the last week of June. I put my traps out. I like to get my zero, but if you get them out by I think June 30th, that's a Monday. And I've also got some corn borer traps going. I would expect in the next week or so in southern Michigan we might see some corn borers flying around.
Okay. Our
Um,
numbers have been pretty low. Is that what you're
yeah.
saying, Chris?
And those of you who have alfalfa, I have found some. Phil said he sees nothing out there, but I did see a field or two that had some pretty high numbers of larvae. And again, sometimes you can just get by with a spray along the tree line where they've kind of come out of their overwintering spot to preserve those beneficial insects. I hate to have you spray. I'd rather have you cut, of course, and which is probably at this point you can probably wait till next week and cut. So that would be the ideal thing. Just check that new growth for the little dudes surviving and climbing up there.
We
I think
put
that's all.
we put slug little traps out, and I have not gotten much around the Shepherd, Michigan area. So,
Okay.
and then there are some little grubs we're seeing in the soil, and I was gonna scoop them up and give them to you here next week, Chris.
Yeah,
So
there's
you
little dribs and drabs, but nothing too exciting yet. And then maybe Marty has an update, so I'll stop.
Marty, do you have anything else you'd like to add?
Yeah, I checked the head scab risk prediction tool and it's showing low risk across Michigan. So that's a good thing. That fungicide, though, at that flowering timing will help to keep that flag leaf clean if any diseases are active. There is quite a bit of powdery mildew around the state.
Mhm.
A little bit of stripe rust that was confirmed apparently in Kalamazoo. I haven't seen any photos or anything to confirm that, but I put that up on the stripe rust map as well. And then there's continued talk about viruses. Chris made a good point this morning when we were chatting. Really good just to get confirmed what you think you might have. And the wheat board is covering testing costs here at the diagnostic lab. So I encourage folk to get a sample in. Even if you think you know what it is, it's not a bad idea to double check.
Yeah, and while this last question gets answered, I'll pull up the link to submitting your wheat for free to the diagnostics clinic. I'll go ahead and put that in the chat box. Okay, Peter, are you able to unmute yourself again here to answer the question about growth regulators?
100%. So
Awesome, thanks.
I, thanks for the question. And so CCC or Cycocel or chlormethoxide chloride is a growth regulator. I believe it's going through the registration process right now in the US. We've had it for a number of years. Both products, whether it's Palisade or we call it manipulator here, would be this chlormethoxide chloride. They both work very, very well. Under adverse weather conditions, we have seen a bit more phytotoxicity with Palisade than we have with the chloride. So I'd give it a little bit of a nod in terms of crop safety, but they both work really well and they're both generally very safe. And we sort of go on price from that perspective.
Awesome. Thank you for being our honorary wheat person as Dennis's computer has failed him. Um, I think, is there anyone else that has anything to add? I know, Jeff, you said that we're not behind in planting, but I would say my farmers would disagree with you.
Well, location. Yeah.
Okay. And with that, I think we can go ahead and wrap it up. I want to thank everyone for getting on for another virtual breakfast, and we look forward to having you on next week for dry beans.